Okay, so let's get started. So welcome to the 2022 flag meeting. It feels so good to be in a room of people. I cannot even tell you. This is the first in-person meeting I've been to in 2.5 years, and I just can't attend another Zoom meeting. I need to see people. I need to see people. So I'm so glad you all are here. Thank you for traveling, and welcome also to the virtual participants of the meeting. I'd just like to start off by thanking our sponsors who are listed here. We're very appreciative of the support so that we can have this meeting. And I want to give a huge shout out to my co-organizers, Justin, Ellie, Sally, and Artyom. If you're all here, could you stand up? Please give them a round of applause. They were a fabulous group of people, hardworking, honest, thoughtful, and it was everything that we decided about this meeting just gelled. We all seemed to arrive at the same choice, so it was really easy. Thank you, GSA. So we make the decisions about the science, but who gets all of this done is the GSA. We're really grateful for them, and they're run by Tracy, who's fabulous. She's not here, but there's a whole group team who makes all of what's happening look very seamless. And we are grateful to each and every one of them. And I would like to ask Susie Brown of the GSA to come and accept these flowers. Susie, accept these flowers. Thank you from this whole team. Thank you. I'd also really like to thank the Fly Board and the GSA DEI committee, who were very helpful in choosing the variety of speakers that we have for you. We have really exciting scientists from diverse backgrounds that we hope reflect the Fly community as a whole. And I'd like to give a shout out to the past, present, and future Fly presidents listed here, Fly Board presidents. We have a great lineup of speakers. So we have a great keynote you're going to hear tonight. We have 12 plenary speakers. We have a ton of platform speakers, workshop speakers, and then all the people who are presenting posters. So thank you all for coming and sharing your science with us. We're really looking forward to hearing from you. We also have a lot of hardworking session co-chairs who worked very hard to select all the platform talks that you're going to hear today. And then some of those session co-chairs are going to coordinate all of the poster judging. So it's a lot of work, and they were all very thoughtful in their choices, and we really are very much appreciate them. We did really well. There's like total registration is, I think, above now 1644. There's about 1,100 people here at the meeting in person, and there's about another 600 online, and we have almost 40 countries. So this is really an international community, and I think it's really great that we're all together. I would encourage you to check out the exhibitors. So you find them next to the poster session, and if you click on this in your app, in your Dros22 app, you can find them. And I'd also like to ask all of you to, if you have the time, to check out the virtual posters as well as the in-person posters. If you've been to the Fly meeting before, The posters are a little different. They're all going to be up one day and then down, and then the next, and then the bees are up and then down. So it's not going to be like it was in the past. And they're also going to be a little bit spaced out to encourage whatever kind of distancing we can get. So please look at the virtual posters. Finally, we ask that you please wear your mask inside. And then also, please consider everybody's comfort level. Some of us, like me, are willing to hug everybody. There are other people who really, don't want to be touched or who just want to have the elbow bump. And there are stickers that some people have. And if they have a particular comfort level and you see that, just please, respect that. And that's it. Enjoy the meeting. And now the current fly board president, who will be current for the next 4 days, Tintin Su, is here to talk to you.
Good evening, everyone. My name is Tintin Su. As Erica said, I'm the president of the Drosophila board, which represents the fly community to further its interests in research, education, and community outreach. I'm here to recognize and respect the indigenous peoples, especially the Kumeyaay, as traditional stewards of the land that we are all on, right? This land right here. We honor these grounds and acknowledge the resilience and strength that all indigenous people have shown worldwide. These will be empty words without accompanying action. The Fly Board continues to strive to improve inclusion and representation in our community activities, including fly meetings like this one. This year, the Fly Board will be adding a new member to represent minority serving institutions and community colleges. We created a new award category to recognize outstanding service to the Drosophila community that will be open to all, not just the PIs, as is traditionally. We generated and voted in a set of good practices that we will encourage future organizers of the fly meetings to follow, including considering all demographic groups when selecting speakers and session chairs. Our goal is to do better each year in being inclusive and representative. I'm here also because we have an obstacle in achieving this goal, namely that we do not have a good idea about who we are. This past year, with help from GSA, we've been looking at all available demographic data for the fly community, and it is incomplete. Most of it comes from surveys like the ones that are sent out after the fly meeting, and the response rate is not great, like below 40%. If we want to make our speakers, our session chairs, our committee members representative of our community, we need more complete data of the community makeup. With this in mind, and with help from the GSA, the Fly Board will be sending out a demographic survey this year. Please, let's all respond to that. And we need to do better than 40%. On that survey, you will have an opportunity to indicate if you represent a traditionally underrepresented group, and if you would like to be involved in community committees and organizing or speaking at fly meetings. This will help generate a database that can be used to make future committees and meetings truly representative. We are modeling it after such a database that the American Society for Cell Biology has generated and is already using. So please respond to the survey, please self-identify, please volunteer to participate, and we must all work together to make the fly community the best it can be. Thank you.
And now we'll hear from Denise Montel.
Well, hello, everyone. It is so good to see you all in person. I'm Denise Montel. I'm a professor at the University of California in Santa Barbara. I served as GSA president in 2020. You all remember 2020, I think. On behalf of GSA, I'd like to welcome you all. GSA is always happy to support the annual Drosophila Research Conference, but this year we are especially excited to be reuniting with this community in person. And to all of you tuning in virtually, we are thrilled that you are able to join us for this amazing meeting from afar. A silver lining of the pandemic is that we figured out how to include the thousands, hundreds, and sometimes thousands of people who want to attend our meeting, but for one reason or another are unable to be here in person. This has always been true, and now we have the technology and the experience to offer the virtual experience alongside the in-person conference. Let's see. We'd like to thank our dedicated organizers. You've heard from Erica Bach, Justin D'Angela, Ellie Hexter, Sally Horn-Badavinick, and RDM Kopp for their work in developing this excellent scientific program. We'd also like to thank the GSA staff for bringing this conference together, COVID curveballs and all. I cannot say enough about the nimble, creative, and dedicated GSA staff. They are a little bit shy, so they didn't want us to put our pictures up here, and I'm really glad that that we gave Susie the flowers. So they wouldn't let us put their picture up here, but Susie Brown and Anne-Marie Mahoney, who are the primary conference organizers, have just gone to tremendous efforts over these past few years that have been, I don't know, challenging seems like an absurd understatement for what the conditions have been that they've been working with. This is one of GSA's first hybrid meetings, and we appreciate any patience you can muster as we do our best to keep events running as smoothly as possible, both in person and online. For those of you who don't know, GSA is an international professional society for scientific researchers and educators in the field of genetics. Many of you probably just clicked join GSA as a member. Click that button in order to save a few dollars on the registration fee, and that's cool. That's reasonable. But what you may not know is that scientific societies in general, and GSA in particular, are an indispensable part of the fabric of our scientific lives. They bind us together when the world seems like it's falling apart. Like, for example, when the in-person 4 years in the making 2020 Allied Genetics Conference had to be canceled due to the pandemic, GSA managed to flip the whole meeting online without any precedent or example or any guide. Just when we were the most isolated, GSA brought us together to network, to share our science, and so that we can continue to make progress despite being locked out of our labs. Conferences are a key contribution that GSA makes, but GSA also works to advance the field through professional development, community programs, advocacy, and publication in its two journals, Genetics and G3, which are published in partnership with Oxford University Press. Genetics is revered for its storied history since its launch in 1916. Genetics publishes papers that advance our understanding of genetic and genomic mechanisms and functions. G3 launched in 2011 as a fully open access journal and is the place for high quality, useful foundational research in genetics and genomics. Both journals are peer-reviewed, peer-edited, and known for their expert reviews, clear decisions, and overall author experience. Manuscripts reviewed at Genetics can be transferred to G3, and G3 editors can often offer a decision based on reviews from Genetics, eliminating the need for re-review. Last year, the GSA journals launched a new neuroscience series, which some of you have already contributed to, and I encourage you to check out. Visit the GSA booth to chat with managing editor Ruth Isaacson and scientific editor Sarah Bay. Chat with real live people who run these journals about the series and to learn more about publishing in Genetics and G3. In addition to being a great place to publish, the GSA journals provide training opportunities for early career scientists. In particular, in the peer-reviewed training program, over 100 students, postdocs, and early career faculty receive formal training in manuscript review at Genetics in just 2021 alone. Applications for the 2022 cycle will open later this year, and we encourage you to apply. In 2020, the Society introduced the Presidential Membership Initiative. This is another program aimed at early career scientists. The PMI program is part of GSA's continuing commitment to improve equity and inclusion in our fields. The PMI offers free GSA memberships, abstract waivers to awardees. It's designed not only to increase the diversity of membership, but also to provide positive experiences for the cohort, a sense of community, and experiences like recognition in genes to genomes, connecting with GSA board members, and participating in networking events. Last year, GSA welcomed 82 inaugural PMI members, and the 2022 cohort will soon be announced. In other efforts to strengthen our scientific community, GSA advocates for shared resources such as Flybase and other model organism databases, whose funding is in jeopardy. 2021 President Hugo Bellin spearheaded a webinar series that drew over 600 attendees and introduced scientists to resources that encourage collaborations between physicians, research scientists, and genome database developers. Currently, GSA's new Public Engagement and Communication Committee is developing educational materials that demonstrate the incredible importance and impact of research using organisms like yeast, worms, flies, fish, and mice. These oral interviews and written stories will be made available to GSA members to use in their own public education and advocacy efforts. So stay tuned to the GSA website and social media channels. Of course, so many of GSA's programs and so much of the advocacy work is in support of students and postdocs. You are our next generation of genetics researchers and educators. On that note, I'd like to turn the podium over to Andrea Day, co-chair of GSA's Early Career Leadership Program, who can tell you firsthand about the opportunities for early career scientists at GSA.
Thank you for giving me this opportunity. It's truly an honor to share with you about the Early Career Leadership Program, or ECLP. The ECLP is comprised of early career scientists from around the world. We meet virtually and work in teams to propose, develop, and implement ideas and initiatives that address the needs and challenges of our community. This platform helps us gain new leadership skills, build a professional network, and demonstrate our abilities through concrete deliverables. The ECLP is powered by 7 subcommittees, each of which consists of co-chairs, members, and advisors who work on various projects and specific goals under the guidance and expertise from subcommittee advisors, GSA engagement, GSA board representatives, and the GSA executive director. I'll briefly outline the main objectives of each of these subcommittees. The steering committee is made up of co-chairs from each of the other subcommittees. Its main purpose is to guide the direction of ECLP, facilitate projects, events, and proposals, and create more avenues for collaboration within the program. The accessibility subcommittee is newly formed, and I serve as the co-chair of it, and also I helped co-found the subcommittee. So let me tell you a bit about my journey. I joined the ECLP in 2020 as a member of the Career Development Subcommittee. While I was actively involved with all the tasks that were assigned to me, on the side, I was always truly very passionate about making science more accessible to students and scientists with disabilities. I had this very naive thought of improving accessibility within ECLP, but had no concrete plan on how to execute it. I approached peers and senior leadership within the program with my idea, and I received an incredible amount of support and guidance from everyone. And through that, I was initially able to build an affinity group with members from all other subcommittees who acted as liaisons. Gradually, we transitioned to establishing a whole new subcommittee, and this year, we successfully recruited new members. Our goal is to understand the access needs of the community and ideate ways to accommodate those through a combination of universal design approaches and increase representation of scientists with disabilities in existing ECLP projects. We make recommendations to members of the ECLP and GSA, such as having closed captions on all our virtual programming, or encouraging people to use colorblind-friendly palette in presentations. Each member of the ECLP can participate in this initiative and gain a new learning experience. This is one of the many ways in which the ECLP has always welcomed new ideas that address diverse needs of the community. The Career Development Subcommittee supports early career scientists by developing career resources, such as workshops and interview series that highlight a variety of career trajectories that are possible with an education and research training in genetics. The community and membership engagement team works towards addressing concerns of the early career scientist community by building resources and communication pieces. The Communication and Outreach Subcommittee aims to bridge the gap between different scientific disciples and also between scientists and the public by crafting a variety of informative articles and managing a weekly newsletter. The Multimedia Subcommittee uses different multimedia platforms, such as podcasts and infographics, to make genetic research more accessible to the broader audience and also promote collaborations in the larger scientific community. The Policy and Advocacy Subcommittee develops resources related to career training policies and helps connect the scientific community with officials and government agencies. Together, we assemble a huge repository of online resources. To represent a few, we have Workshop Wednesday series and Decoding Life interview blogs that demonstrate the diversity of PhD careers. We also have Genetics in Your World podcasts, weekly newsletter pieces, and articles in Genes to Genome that either communicate recent research, provide professional development opportunities, or help increase awareness about important issues. Along with that, the ECRP also offers us trainings that are facilitated by professionals and experts. For example, we have a 4-week online science writing workshop. We also participate in a 6-week leadership training program to learn about team building and management. Also, the ECRP and GSA gives us the opportunity to participate, co-host, or organize events at GSA conferences, both virtual and in person. Many of you may have attended some of these either earlier today or in our virtual programming. The ECRP provides students and postdoc members from around the world with professional training, leadership skills, and rewarding experiences that enact change and advance their careers. Thank you.
Thank you, Andrea. For those of you interested in learning more about the Early Career Leadership Program, Jessica Velez, who is GSA's Early Career Programs Manager and a former ECLP participant herself, will be at the GSA booth tonight. So please stop by and meet her. Now it's my pleasure to welcome Alyssa Armstrong, who will be introducing this year's Larry Sandler Award winner.
All right, good evening, everyone. I am Alyssa Armstrong, an assistant professor at the University of South Carolina, and I had the distinct pleasure to chair the Larry Sandler Memorial Award Lecture Committee. So this award was established in honor of Dr. Larry Sandler by his colleagues, friends, and students, and it serves to honor his contributions as a Drosophila geneticist, some of which are listed here, particularly in the area of meiotic drive, but also to honor his legacy as an outstanding mentor. I recently read personal reflections about Larry Sandler by one of his colleagues and friends, Dan Lindsley. And from reading that, what stood out to me was that he seemed to be a scientist for science's sake. And that's something that I really take to heart. Each year, the award is presented to an outstanding PhD graduate. This is based on nominations from advisors as well as their dissertations. So I'm going to take a moment to encourage all advisors to really think about their outstanding graduate students and the work that they've done to consider submitting nominations for them toward the end of the year. This year's awardee will join this prestigious list of researchers and a committee of myself and 4 other excellent faculty members, Sophia Arujo, Amanda Laraquente, Luciano Matskin, and Timothy Mosca, all served on this committee to really hammer out this pretty difficult task. There was an outstanding pool of PhD nominations. And so we had lots of thoughtful, careful discussions about these candidates. So I want to take the time now to recognize the 3 runners-up, Dr. Maureen Lamb from the Tootle Lab, and her dissertation was entitled Defining the Functions of the Actin Bundling Protein Fasten in Invasive Collective Cell Migration. Dr. Luis Hernandez Nunez, whose dissertation was entitled Systematic Characterization of Sensorimotor Transformations in the Drosophila larva, and Dr. Cheng Liu, whose dissertation was entitled Building an Allocentric Traveling Direction Signal via Vector Computation. So now I want to take the time now to present this year's awardee, Dr. Lianna Wat, who was a graduate student in the lab of Dr. Elizabeth Rideout at the University of British Columbia. And so now she's going to tell us about her elegant studies in understanding the sex-specific regulation of fat metabolism in Drosophila. And Lianna, welcome.
All right. Good evening, everyone. So thank you to the organizing committee and to the awards committee for having me here today. And I'm really excited and really honored to be giving the Larry Sandler Memorial Lecture tonight. So before I begin, I also want to acknowledge that I did my PhD at the University of British Columbia, which is situated on the traditional, ancestral, and unceded territories of the Musqueam people. And doing my PhD here is a privilege for which I'm really grateful. So in most animals, maintaining energy homeostasis is crucial to survival. What this means is that during times of nutrient abundance, animals need to store excess energy so that during times of nutrient scarcity, they can mobilize these energy stores to maintain basic biological and cellular functions. So the main form of stored energy in most animals is actually in the fat, and typically in the triglycerides. So while many things regulate triglyceride or fat homeostasis, one key regulator is actually biological sex. So in this paper here by Saber and Spradling in 2015, they demonstrate that in mated females represented in white, actually store more fat compared to males represented in this black bar. So while this is a really exciting and really interesting phenotype, we actually know very little about the molecular mechanisms that underlie how females store more fat compared to males. So this is really at the core of the interest of my PhD project. I wanted to know what the metabolic genes and metabolic pathways are that are contributing to these sex differences in fat metabolism. So when I started my PhD, one of the first things that I did was to characterize the sex differences in fat metabolism in Drosophila. So here I'll start with the fat storage graphs. So on these y-axis, this y-axis represents the percent body fat. And for this portion of the talk, the orange represents females and the blue or purple represents males. So what you can see here is that we see in virgin females that they also store more fat compared to males. So in addition to looking at fat storage, I also looked at fat breakdown. So to measure fat breakdown, we calculated the change in percent body fat represented in this y-axis between the hours of 0 and 12 hours post-starvation and 12 and 24 hours post-starvation. What you can see here is that in response to starvation, males lose a significant amount of body fat between both of these time points, but females actually do not lose a significant amount of body fat between either of these time points. So what this data is demonstrating to us is that females have increased fat storage and a delay in fat breakdown compared to males. So I was interested in understanding what the metabolic effectors are underlying these sex differences in fat metabolism. So we decided to start by using qPCR to measure the mRNA transcript levels of a host of lipid metabolism genes. So we looked at lipid synthesis genes, lipid storage genes, and lipid breakdown genes. So this y-axis represents the sex difference in gene expression. Anything colored in blue represents something sex-specifically regulated in the males. Anything in orange represents something that was significantly female biased. So what you can appreciate from this graph is that there's actually widespread sex differences in the regulation of these lipid metabolism genes. but we decided to focus on one gene in particular, this one right here called Brummer, because it had a really unique gene expression profile. So I'm showing you this gene expression profile here in a radar plot. So this y-axis represents the fold change in mRNA transcript levels. And starting at the top, moving clockwise around is increasing time points post-starvation. Hopefully what you can appreciate is two different things. One is that at all time points post-starvation, Brummer transcript levels are male biased. But what made this gene stand out to us was that in response to starvation, males really immediately and really dramatically increase their transcript levels, whereas females have a much more blunted and delayed response. So Brummer is a triglyceride lipase that is responsible for breaking down triglycerides into diglycerides, so catalyzing this rate-limiting step of fat breakdown. So because it's such an important enzyme in the lipid metabolism pathways, we actually know quite a bit about what Brummer does in regulating fat metabolism in general. So from the literature, we know that high levels of Brummer lead to a less fat phenotype, whereas low levels of Brummer lead to a more fat phenotype. So combining this information with what we now know about the sex difference in fat storage, our hypothesis is that high levels of Brummer in males is what leads to their less fat phenotype, whereas low levels of Brummer in females is what leads to their more fat phenotype. So to test this hypothesis, we decided to start by looking at animals with whole body loss of Brummer. So here in our lighter colors are our controls, and you can see females storing more fat than males. But in our darker colors, if we look at animals with whole body loss of Brummer, we now see that the sex difference in fat storage is abolished. And this is due to a male biased increase in fat storage. So in addition to fat storage, we also looked at the fat breakdown phenotype. So in males with whole body loss of Brummer, they are now no longer losing a significant amount of body fat between either of these time points post-starvation, similar to what we see in females. And in fact, we see no effect of loss of Brummer in female fat breakdown. So together, this demonstrates that Brummer is actually required for the sex differences in both fat storage and fat breakdown. Now, because we've used whole body mutants for Brummer, our next question was to try and identify the anatomical focus of Brummer's effects on regulating the sex differences in fat metabolism, because we know that Brummer is actually expressed in many different tissue types. So we decided to use the UAS-GAL4 system to use an RNAi line to inhibit Brummer function in specific cell types and specific tissue types. So we looked at the gonads, the fat, the gut, the muscle, and the postmitotic neurons in the brain. So we actually found two hits, one in the somatic cells of the gonads and one in the postmitotic neurons of the brain. But given our time constraints, I will be focusing on one of these today, and that will be in the neurons in the brain. So if anyone wants to ask me about the somatic cells of the gonad later, I would be happy to chat about that. So what we found was when we inhibited Brummer function in the neurons, that there was no effect on fat storage, but there was a male-specific effect on fat breakdown. So specifically, loss of Brummer function in the neurons led to these males no longer losing a significant amount of fat between 0 and 12 hours of starvation. And this is a really interesting and really exciting finding for us, because we didn't expect a lipase to have a function in the neurons in regulating the sex difference in peripheral fat metabolism. So this led us to wonder what Brummer is doing in these neurons to actually regulate this sex difference. So we know that Brummer is a lipid droplet associated protein. So this means that it sits on the surface of lipid droplets to regulate triglyceride breakdown. So we were interested in looking at the lipid droplet dynamics in these neurons, but as many of you may be able to appreciate, looking at lipid droplets within neurons is a really difficult task. So we were very lucky to be able to collaborate with Dr. Michael Welty, who generously gifted us this UAS lipid droplet targeted GFP line. So essentially how this tool works is you use the GAL4 system to drive expression of the lipid droplet targeted GFP specifically in a cell type, and we did the neurons, and this will mark these lipid droplets with a GFP halo. So we first decided to test this in our animals to see if this actually works. And here in these green punctae, you can see that these are marked by the lipid droplet targeted GFP. So we simultaneously stain these brains using a neutral lipid dye called Lipid Tox, and this will stain anything that is a lipid droplet in magenta here. So what you can appreciate are these white punctae that are positive for both the lipid droplet GFP and the lipid tox, demonstrating that these are indeed lipid droplets within neurons. So now that we've confirmed that this tool works in our hands, we wanted to look at lipid droplet dynamics or lipid droplets in the brain of males and females. So here on the top is a picture of a female brain, and on the bottom is a picture of a male brain. So hopefully the resolution is clear enough for you to see that there are these individual punctae throughout the brain, but they kind of localize to these regions, like perhaps here in the optic lobes and here in the mushroom body. So going forward, this is actually the first time we've been able to see lipid droplets within neurons. So this is a really exciting finding. And going forward, the lab is now really interested in using this tool to understand how Brummer is regulating these lipid droplet dynamics, what the sex differences are in these lipid droplet dynamics, and perhaps how Brummer is regulating those. So now we have this body of really interesting data on the sex-specific regulation of Brummer. But what we were really now curious about is what are the physiological consequences of this? Why is there a sex-specific regulation of a triglyceride lipase? So we know that fat metabolism and Brummer has actually been linked to many life history traits, things like lifespan, fertility, and starvation resistance. So we decided to look at these three phenotypes in our flies in both males and females. So we decided to start by looking at animals with whole body loss of Brummer again. So here I'm going to show you some lifespan data. In this y-axis, these are the percent alive animals, and the x-axis is their age in days. So in our two lightest lines here, these are the control female lifespan and the control male lifespan. And you can see this really nice sex difference where females live longer than males. But what's really interesting is that animals with whole body loss of Brummer no longer have this sex difference in lifespan due to a female-specific decrease in lifespan. So this is a really interesting finding for us because if you'll recall a couple slides earlier, loss of Brummer in the whole body actually had these really large effects on male fat storage and not female fat storage, yet it has these massive effects on female lifespan and not male lifespan. So this is another avenue that the lab is interested in pursuing. We also looked at fertility in males. So in this graph, this y-axis represents the number of progeny produced from a 24-hour mating period. So we used Brummer mutant males and mated them with 3 wild-type females. And what you can see here is that there's no bar for the Brummer mutant males, and that's because virtually none of them were able to produce any progeny. So this indicates to us that Brummer function is playing this really key role in regulating male fertility, which is another avenue that the lab can now pursue. The third phenotype we looked at is also starvation resistance. So in this case, the x-axis is hours post-starvation. And again, in our lightest colored lines, you can see this really nice sex difference in starvation resistance, where females are able to survive starvation longer than males are. And what's interesting is that loss of Brummer function in the whole body also abolishes the sex difference in starvation resistance due to this male-biased increase in starvation resistance. So altogether, what these set of data is telling us is that the sex-specific regulation of Brummer plays this really important role for regulating these sex differences in different life history traits. So in this project, we actually demonstrated that there is a widespread sex-specific regulation of fat metabolism genes, and we specifically identified a novel function for Brummer or ATGL in regulating the males acting in the male somatic cells of the gonad and neurons to regulate the sex differences in fat metabolism. Additionally, we demonstrated that lipid droplets are actually present in neurons under normal physiological conditions. So this project was wrapped up in about 2020, and we published this in PLOS Biology. But one thing you may have noticed is while Brummer explains quite a lot of the sex differences in fat metabolism, it didn't explain all of them. So after we published this work, we pivoted our focus to look more upstream at the sex determination pathway and also more broadly at metabolic pathways to try and understand what else is contributing to the sex difference in fat storage. So before I continue to this portion of my talk, I just want to give a brief overview of how sex is determined in flies. So sex is determined in flies based on the number of X chromosomes. In females, they have 2 X chromosomes, which allows for the production of a functional sex-lethal protein, which allows for the production of a functional transformer protein. And this governs female development and female behaviors. In males, they only have 1 X chromosome, so they have no functional sex-lethal, no functional transformer, and this allows fruitless and double sex to govern male developments and male behavior. So the important thing to remember from this slide is that females have a functional transformer and males do not. And we decided to focus our studies on transformers' effects on fat metabolism in males and females because it's the most upstream sex determination factor that we can manipulate without causing lethality. So our first question was to determine whether transformer is a necessary regulator of the sex difference in fat storage. So we decided to do this by looking at females with whole body loss of transformer. So for this portion of the talk, females will remain in orange, but males will now be in burgundy or red. So here in this fat storage graph, you can see that females with whole body loss of transformer have significantly lower fat storage compared to control females. We also did the opposite experiment where we used the UAS-Gal4 system to overexpress the female isoform of transformer ubiquitously. So when we do this in males, you can see that they have this male-specific increase in fat storage compared to their controls. So this set of data is demonstrating that transformer is indeed a key regulator of the sex difference in fat storage. Now, similar to the Brummer paper, we used whole body gain and loss of transformer. So our next question was to identify the anatomical focus of transformer's function to regulate the sex differences in fat metabolism. So we decided to overexpress a transformer in specific cell types and specific tissues to feminize them. So we expressed TRAF in the gonads, the fat, the gut, the muscle, and the brain. And really interestingly, when we feminized the postmitotic neurons of the brain, this actually led to a male-specific increase in fat storage. Now, as you may know, there are many different neuron subpopulations that make up all of the postmitotic neurons in the central nervous system. So we actually then took this one step further and did a secondary screen where we feminized different subpopulations of neurons. And we found one neuron subpopulation that played a role in regulating the sex difference in fat storage. And that was in the AKH producing cells. So when we feminized the AKH producing cells, or the APCs, this led to a male-specific increase in fat storage. And again, it had no effect in females. So what is the AKH producing cells? So they produce a peptide called the Adipokinetic hormone, or AKH, and this is actually a key regulator of fat metabolism. So we know that AKH plays this role in regulating fat metabolism in general, and from the literature, we know that high levels of AKH lead to a less fat phenotype, and low levels of AKH lead to a more fat phenotype. So after we found transformer function in the APCs, our first question was to see whether the AKH pathway is sex-specifically regulated. So we did this by looking at 3 different traits. The first thing we looked at was the AKH mRNA levels. So what you can see here is that AKH mRNA levels are higher in males compared to females. And this is also true when we look at the AKH receptor mRNA transcript levels. The second thing we looked at was the activity of these neurons. So we used a calcium-dependent reporter of neuronal activity and expressed it specifically in the APCs and imaged them. So what you can see here is that female APCs have very low levels of activity, whereas male APCs have very high levels of activity. So in addition to looking at the APC neuronal activity, we also looked at a protein readout of the AKH signaling pathway called phosphorylated IRE1. So here in this Western blot, you can see that males stain much higher for phospho-IRE1 compared to females. So putting all of this data together, this really demonstrates to us that the AKH signaling pathway is higher in males than in females. And putting this information together with what is known in the literature about AKH, this is our hypothesis. High levels of AKH signaling in males leads to their less fat phenotype, whereas low levels of AKH signaling lead to their more fat phenotype in females. So in order to test this hypothesis, we actually used many different genetic techniques to inhibit AKH signaling activity in males. So here I'm demonstrating 2 different experiments we did. In this one here, we used an RNAi line to knock down AKH expression specifically in the APCs. And here we see this male-specific increase in fat storage. Next, we also used an inwardly rectifying potassium channel to inhibit the neuron function of these APCs. And we also see this male-specific increase in fat storage. In addition to these techniques, we also ablated these APCs and looked at whole body mutants for AKH and AKH receptor, and we see the same male-specific effect on fat storage. So this really solidifies that the sex-specific regulation of AKH signaling is contributing to the sex difference in fat storage. So now that we know this about the AKH signaling pathway, and we know that transformer is playing a key role in regulating the sex difference, our next question was to ask, whether transformer is regulating the sex difference in fat storage through the sex-specific regulation of AKH. So to answer this question, we decided to perform an epistasis experiment. So here in this case, these light gray bars are our control females, and we looked at these females with whole body loss of transformer, represented in these two dark gray bars, or circles, and you can see that these females have a decrease in fat storage. And what was really exciting was that when we simultaneously ablated these APCs, this actually rescued the female fat storage phenotype back to that of a control female. So this experiment really demonstrates that the transformer is indeed regulating the sex difference in fat storage through its sex-specific regulation of AKH signaling. So again, we were really excited by all of these findings, and we were always asking this question, like, why is there a sex-specific regulation of AKH signaling? So to answer that question, we again looked at these physiological consequences. So we know that their AKH signaling is associated with regulating things like feeding behavior, lifespan, and fertility. So we did look at all three of these phenotypes in our animals, but I'm not showing you any feeding behavior today because in our hands, inhibition of the AKH signaling pathway had no effect on feeding behavior in either sex. So let's start with the male data. So we decided to look at males with whole body loss of AKH, and we first measured mating behavior. So we introduced one male to a group of 3 wild-type females, and for the first hour, we scored these males every 10 minutes on whether they were able to successfully copulate with a female or not. So that's represented here in this y-axis as the proportion mating. So what's really interesting is that in pink is our control males, and you can see that right off the bat, almost all of them are able to successfully mate with a female. But males with whole body loss of AKH have the opposite effect, where most of these males are not successfully mating. So this demonstrates that AKH is responsible for some parts of male mating behavior. After this mating assay, we removed the males and allowed the females to lay eggs, and we counted the number of offspring after 10 days. So here in this graph, you can see that males with whole body loss of AKH produce significantly fewer offspring compared to control males. So this set of data demonstrates one reason why males have higher levels of AKH signaling, and this is because it's actually necessary for them to maintain their normal mating and fertility phenotypes. So what about the females? So here in the females, we again looked at animals with whole body loss of AKH, and we see that females with whole body loss of AKH actually produce more offspring compared to their controls. And this is also something that we found really interesting. And from the literature, we know that when female fertility or fecundity is increased, this is often accompanied by a consequence in another life history trait, like lifespan. So we also looked at female lifespan. So here we have females with inhibited APC activity, and you can see in this orange bar here that they have a significant decrease in their lifespan compared to their controls. So together, this set of data gives us one reason why females might have lower levels of AKH signaling, and that's because it's actually even more beneficial for their fertility. So altogether, here I've demonstrated a novel role for the sex determination gene transformer in regulating the sex differences in fat metabolism through the AKH signaling pathway. I also demonstrated that the AKH pathway is sex-specifically regulated at many levels. And this sex-specific regulation of the AKH pathway allows males and females to develop distinct lipometabolic profiles that favor reproductive success in each sex. So this project was wrapped up earlier this year, and we published it in eLife. So I hope you can go check out these 2 papers later on. But overall, at the outset of my PhD, the goal was to identify metabolic effectors that are contributing to the sex differences in fat metabolism. So I identified a sex determination gene transformer having one of these roles, a metabolic gene Brummer for having a role, and also a metabolic pathway, the AKH pathway, in contributing to these sex differences in fat metabolism. So altogether, I hope that one of the major takeaways you have from my PhD is the importance of studying both sexes, because by studying both sexes, not only do we understand more about how fat metabolism is regulated, but by comparing males and females, there are some things that you can only see when you compare the 2 sexes. So altogether, I really want to thank the lab I did my PhD in, particularly my supervisor, Liz Ryda, because she's been an outstanding support system for me for the last 5 years. Also, all of the people in the lab who contributed to these 2 papers, like Jason, Charlotte, Jahid, Pooja, George, and Colin, as well as my wonderful committee members, the collaborators that made these projects possible, and all of our funding agencies. So since I finished my PhD, I've actually moved on to start my postdoc. So I'm currently a postdoc at Stanford, working in the Svensson lab, where I'm continuing to study the sex-specific regulation of energy metabolism in a mammalian system. So I'm only taking a break from the fly work for a couple years, so don't judge me, I'll come back for sure. But with that being said, I'm really grateful to be here. I'm really happy to share my work with you, and I'd love to take any questions.
Hi. Hi, great talk. The males with AKH knockdown had fewer successful mating events, but did they have the same number of progeny per successful mating event as the normal males? Or was their fertility also reduced as well as their ability to mate?
So their fertility after 1 hour of mating was reduced. So that corresponds to the mating behavior data you saw. But we did find one really interesting thing. So if we increase the mating time to 24 hours, the difference between the number of progeny produced is actually not there. So we think that it's probably an effect on the male mating behavior and not so much like perhaps spermatogenesis.
Hi, Lianna.
Hello.
Beautiful talk.
Thank you.
Do you think connecting the first and the second half of your talks is AKH controlling Brummer? And if so, how are the neurons, the APC actually, are they controlling Brummer in the targets where Brummer is differentially regulated in the sexes?
So I was really hoping somebody would ask me this. So we think that they might be they're working together. So we actually performed an experiment where we we knocked out AKH receptor and Brummer in the whole body together. And you can see that loss of each of these individually in the males leads to a small increase in fat storage, but losing both leads to this further increase in fat storage. So we think they're working in parallel, but we have to check to see if Brummer transcript levels are in these AKH neurons and if they are regulated.
Thank you.
Okay, great. There's one question from the chat from Rodrigo Nunez. Great talk, Lianna. Considering the published results of the Cochem group DAM 2020, do you know if these males with Brummer knockdown have a dehydration phenotype and can be rescued by giving them water?
That's a really interesting question. So I guess we're not sure if it's a dehydration phenotype, but these flies are likely not dehydrated. So when we starve them, we put them on, actually I didn't talk about starvation, did I? So I'm not sure actually, I guess the answer is I'm not sure.
Let's thank Dr. Watt.
Thank you.
So I'm Ellie Heckscher. I'm a assistant professor at University of Chicago, and I'm a co-organizer of this event. Before I forget, I wanted to make an announcement that if anybody would like more space, there is ballroom B over here that is also available, and I think the talks will be projected there as well. So if you feel like you need room, please take advantage. Okay, so I am here to introduce our keynote speaker. So our keynote speaker was my postdoctoral mentor, full disclosure, Dr. Christo from the University of Oregon. And so, you know, Chris has been decorated for many, many things. I think probably one of the ones he's most proud of is being elected to the National Academy, I think in 2017-ish. He's been a Hughes investigator since the 90s. And I personally sort of consider Chris to be one of the fathers of modern Drosophila neurobiology. He was there when the transition was happening from beautiful embryology and locusts to starting to bring molecular tools to bear on the process of asymmetric cell division, cell fate specification in the nervous system. And now he's gone on to do work that is very close to my heart, which is how stem cells populate circuits. And then the last thing I just wanted to say about Chris is that he's a fabulous mentor, and he's, I think, mentored over 20 postdocs, over 30 graduate students. I have no idea how many undergraduates and high school students in the lab, and he's, you know, wonderful member of the community. So I had a lovely time in his lab. I learned a lot, and it is my absolute pleasure to introduce him. So welcome, Chris. Oh, one other thing, sorry, before you do, we will hold Chris's questions and just go straight to the mixer, and you can ask him all the questions you want there. So when he's done, you're done, yeah?
Thanks, Ellie. And I definitely do not want to be between you and the beer. So we'll do questions out there. And if somebody knows how to switch the slides over, I will take help. Refresh button. This. Ah, wow, that's cool. Yeah, very nice. Yeah, while it's opening, I want to also echo the previous people that have been up here and say how fantastic and marvelous it is to see people in person, to see maybe not their chins yet, but at least their eyes in person. And I know I have had a real boost in my mental health over the last 6 hours I've been here. You know, just talking to people, this is amazing. Yeah, so we had a hard time in our lab with COVID, like almost all of you, I'm sure. And we tried various ways of cheering each other up. And one of the ways we found was pretty effective was to rent out a movie theater and have our whole lab pod go see a movie together. This happens to be the movie Mandibles, which is a terrible title, but it's about these two slackers in France who find a dog-sized fruit fly and train it to go get groceries so they don't have to pay for their groceries. And you all know that flies have motion bodies for learning and memory. They're easily trained. They have a central complex, which allows them to do complex navigation behavior. And so the movie really was, you know, if I was the scientific advisor, I'd give it 2 thumbs up. And I highly, this is the audience for that movie. It's, as one of my students said, it's dumber than Dumb and Dumber, which is true. But this audience, if anybody, would appreciate it. So there's a tip. Okay, I'm going to talk about 4 people's work today. A tremendous postdoc, Sonia. I'll tell a vignette about her work. A tremendous graduate student, Austin, will be the second vignette. And then Brandon, and now Heather taking over from Brandon, will be the third one. So if the first, if you find you're not tracking in the first story, I'll tell you, and we'll start the second story, and you can jump back in again. Okay, what I'm going to tell you about is our lab's efforts to understand the generation of neuronal diversity, and more recently, how that leads to circuit formation, and focusing on spatial and temporal factors. And there's 3 different mechanisms that the field, meaning me and 100 other labs, the field has built consensus on the 3 mechanisms that give rise to diversity within the larval nervous system. That first instar, or I'm not sure what instar that larvae is, but the CNS is the brightly colored object in the multicolor flip out, or MCFO stain done by Lorena Manning, a tech in the lab. You can't see the 2 brain lobes, and I'm not going to talk about them. I love those brain lobes and the projections into the brain lobes, and I'm curious about what they're all doing. But we're keeping our focus on the ventral nerve cord, segmentally reiterated, 3 thoracic, 8 abdominal segments, as you know, bilaterally symmetric. The unit of development then is the hemisegment, and there's 300 neurons in a hemisegment. So basically an entire nematode's nervous system, 300 neurons in each hemisegment. And all of those 300 neurons are morphologically distinct by EM reconstruction and molecularly distinct by, as far as we can tell with all the antibodies and probes we've used. So 300 diverse neurons that establish the basic motor program of the larva. How do they come there? How do you get 300? And so the next 3 slides will show you 3 mechanisms built up over the years from the field. The first mechanism is spatial patterning of the progenitor population called neuroblasts. Neuroblasts are arrayed in an orthogonal pattern, and famous patterning genes like in Grail, gooseberry, hedgehog, and columnar genes, VNDI and the MSH, create an orthogonal tartan-like pattern. And the combination of row and column genes creates neuroblast identity. And each neuroblast is unique in its molecular identity and the clone of neurons it will produce. So the first step is making 30 neuroblasts from the beginning of embryogenesis in the neuroectoderm. So that's 10% of the way there, 30 neuroblasts, 300 neurons. What's next? What's next is called temporal patterning. Each neuroblast sequentially produces a cascade of transcription factors shown there: hunchback, Krupel, PDM, Cas. Today we're going to focus on hunchback to keep a common thread. All three stories I'm going to tell you star hunchback as its a major player. It controls the identity of the firstborn or secondborn neurons. Each neuron, each daughter cell that's born from hunchback positive neuroblast will inherit hunchback up until it's postmitotic, and then typically hunchback disappears, so it's transient. If the stem cell makes it to the Krupel window and it gives rise to a daughter cell, that will inherit Krupel, and so on and so on. And so there's an average of 5 divisions per neuroblast. So 30 neuroblasts, 5 progeny, that's 150 neurons. And I'll focus more on the second step in a minute. But now the final step to go from 150 to 300 is every single one of those daughter cells divides asymmetrically to form a daughter cell that inherits NUM, which is a Notch inhibitor. And so the blue cells are NUM positive and Notch negative. The red cells express Notch target genes like HAY. And so this diversifies the neurons, the nervous system twofold from 150 to 300. And you may say those two neurons that are siblings may not be very different, but in fact, that notch on, notch off difference is profoundly important. And I'm not going to talk about it anymore today, but all of the notch on daughter cells project to the motor neuropil. All the notch off daughter cells project to the sensory neuropil. So it's a major distinguisher, but not the topic for today. Okay, and like I said, I'm going to talk about 3 stories. The first one is how spatial and temporal factors are integrated to specify neuroblast-specific neurons. How does hunchback acting in, say, neuroblast 7-1 produce a motor neuron? How does hunchback acting in neuroblast 7-4 produce an interneuron? How are those spatial factors and those temporal factors integrated to create a specific progeny identity? That's the first vignette. And then I'll talk about 2 stories about how hunchback, we're trying very hard to understand not just its role in molecular identity, which is what it's well characterized for, different transcription factors in neuropeptides and neurotransmitters, but connectivity, morphology and connectivity. I'll tell you a story about 2 different neuroblasts, neuroblast 7-1 and neuroblast 5-2, and how we're learning the role of hunchback in specifying connectivity in circuits that these progeny assemble. And that's why hunchback is over there on the far. Just a reminder, I don't think anybody in this audience needs a reminder of hunchback probably. Zinc finger protein, famous for its role in AP patterning, and also reused in the CNS to initiate firstborn identity in all of the neuroblast lineages. So, you know, as an embryo, you can't probably get much more important than hunchback. Okay. And to highlight the beginning of the first part of the talk, how does spatial and temporal patterning integrate? I just want to highlight 5 different lineages where hunchback has been studied. The firstborns or secondborns are hunchback positive, and the names of those neurons are listed there. If we take out hunchback, you lose those neuron identities. We don't know what happens to them. They lose all their molecular markers. We can't track them. They're not there. If you misexpress hunchback, all of the neurons that follow are transformed into early born fates. So it's very important for specifying those fates. And if you look at hunchback in different neuroblasts, you'll see each neuroblast uses hunchback to make a different type of neuron. How does hunchback, this one transcription factor, create a different neuron in each of those stem cell lineages? And how are those integrated? And that's what I'll tell you about. It's the work of Sonia Sen, an amazing postdoc who came into my lab with so much joy, energy, and enthusiasm, and took on this very, very difficult project and did an amazing job. It's one of my top 3 papers I've made in my entire career. I'm very proud of her and of the work. And so the neuroblasts that she's focusing on in her project are neuroblasts 5, 6, and 7-4, which are adjacent neuroblasts. They make totally different firstborn cells. And she wants to ask, how does that happen? And you can imagine that's a very difficult technical question to answer. And she made use of 2 things. One is specific GAL4 lines that label just 5, 6 or just 7, 4. So she now has genetic access to each of those lineages. That was very important. And those lines were developed by Haluk Lasin and Jim Truman's lab and by Seven Lai in my lab. And they've been essential for everything I'm going to tell you about is based on GAL4 lines that label specific lineages. And so she had 2 GAL4 lines and asked the question, how does hunchback create 2 different progeny in 2 different neuroblasts? And there's 2 models that she thought of. The sort of obvious 2 models. One is that it's a combinatorial code where hunchback acts with different spatial factors to create different output. Very simple combinatorial code. May seem like that's the most likely mechanism, but those spatial factors go away pretty quickly. And so that combination, the very simplest model, is not so likely because by the time hunchback is present, the gooseberry and grail are gone. But they may have downstream genes that take their place. So it's still a viable model, this combinatorial code. The other is that the early acting spatial factors create chromatin domains, sculpt out either open or closed chromatin domains that are different in each neuroblast. So each neuroblast has different chromatin. Therefore, hunchback is biased in its binding and has different binding sites. So to test these models, what Sonia did was use the DAM ID method developed by Steve Henikoff first at the Hutch and by Andrea Brandt and Tony Savell at Cambridge. And I'll tell you a little bit about it on the next slide. But the bottom line is UAS DAM methylase will detect open chromatin in a genetically tractable way. So we can drive it in whatever lineage we want and look for open chromatin. And that would distinguish the 2 models. If there's the same chromatin in both neuroblasts, it would favor model 1, the combinatorial code. If the chromatin is different, of course, it would favor the chromatin model. And then she also made a DAM hunchback transgene, which would detect hunchback binding sites, as I'll show you on the next slide. So this method is really elegant and it's worked well for us. At the time Sonia started this, it had only been used on many fold larger numbers of cells, and the labeling was done for 24 or 48 hours. So it was a challenge for her working with Tony Savell, who is a collaborator of ours on this project. Challenge of hers to work it down to get just a few hours of embryogenesis and then just to get a few cells. So the input was very, very different from what had been published. And so if you look at the little diagram on the upper left, the transgene is a interesting multi-open reading frame transgene. It has UASs, so it can respond to GAL4. The first ORF makes the reporter cherry. That's to validate that we're getting neural S56 specific expression or neural S74 specific expression. And then there's transcriptional stop codons. And then only in transcripts that read through into the second open reading frame will make vanishingly small levels of this DNA methylase, DAM. And that's necessary to keep the level so low that it won't be toxic. And I'm not going to tell you the controls on toxicity, but the flies, when you misexpress it pan-neuronally, they're perfectly fine and they hatch out and they're fertile and everything is great. So the minimizing DAM levels works. DAM binds wherever there's open chromatin, surveying the genome. It methylates at GADC sites. There's very, very little adenosine methylation in Drosophila, so the signal to noise is quite good. And then you're able to digest with DPN1 on those sites and sequence the fragments, and you have an idea of where the chromatin is open after sequencing. A very, very similar approach is used to map hunchback binding sites in these lineages. A fusion protein between DAM and hunchback allows hunchbacks to find its binding sites and then methylate it around in the neighborhood. And so we can use this to identify the hunchback binding sites. So with these 2 tools, bless you, with these 2 tools, we are able to look for open chromatin and hunchback binding and clearly distinguish these models if the experiments work technically. Now, DAM alone has been used by Tony Savell and Andrea Brandt for a few years now, and it's been well characterized. No problem. We did the controls also, but I'm not going to show them to you. What's new for Sonia was this DAM hunchback fusion protein. So we wanted to make sure it was working properly. The title of that says DAM hunchback binding is reproducible. Right under the green box, it says stage 9 whole embryos. We're not really at the experiment yet. We're just at the testing the reagent point. So if you look at the heat map, it's highly reproducible in its binding. This is the hunchback binding from the DAM hunchback fusion protein. Very reproducible, 3 biological reps. 700 kb of DNA shown on the right there. And you can just see qualitatively that the binding sites are very, very reproducible. So DAM hunchback binding is reproducible. It works. It's a robust mechanism. And it matches the published DAM chip data. Again, from whole stage 9 embryos. Lee et al. Did chromatin immunoprecipitations. When we compare their data and our data, we see a very nice correlation of hunchback chip and DAM hunchback binding. That's the big blue peak in the bottom left. And then just again, looking at it qualitatively, the hunchback chip and DAM hunchback binding are very, very similar. So these techniques are reproducible, they are viable, and they are potentially good enough to use on just a few neuroblasts, which is what she had to go next. So from whole embryo to individual neuroblasts, oops. Here's the GAL4 lines, neuroblast 5-6 specific on the top, neuroblast 7-4 specific on the bottom. GAL4 is expressed in the neuroblast and its immediate progeny. We know hunchback can create firstborn fates for about 5 divisions before it cannot anymore. And that's about 3 hours. And so that's the window that Sonia did the expression. She aged them for 3 hours from neuroblast formation to 3 hours later. And then either did UAS DAM to see the chromatin state or UAS hunchback DAM to see if hunchback binding was different. And the next 2 slides will be those 2 results. First, neuroblast 5-6 and 7-4 have different chromatin landscapes. I'm showing you the heat map on the upper left. You can see neuroblast 7-4 for 2 replicates in this case has quite good reproducible alignment from 2 different biological reps. But what's really important is that when you compare neuroblast 7-4 to neuroblast 5-6, there's almost no correlation. It's very low correlation coefficient. That means there's very little in common. That means they have different chromatin. And the visual pileup of the counts is shown to the right. Some genes like engrailed are closed chromatin in 5-6, open chromatin in neuroblast 7-4 shown in green, but other loci are open in neuroblast 5-6, rutabaga and pebble, for example, and their genome in just the next neuroblast over, the neighboring neuroblast, their genome is closed. And the volcano plot shows about 3,000 genes that are differential, or not genes, but loci, regions of the genome, 3,000 regions that are differentially open in the 2 neuroblast lineages. So really massive chromatin differences in adjacent neuroblasts. Okay, what about hunchback binding? And the same thing is observed with hunchback binding. Again, in the heat map, 3 replicates in this case. Very good correlation coefficients between 7-4 and 7-4. But when we look at the 2 neuroblasts, 7-4 versus 5-6, we see very low correlation coefficient. So hunchback binding is different in the 2 neuroblasts. Again, the examples on the right, hunchback is not binding at the engrailed locus, but it is at rutabaga and pebble in blue. I'm sorry, in gray underneath the blue. So the blue is the open chromatin. The hunchback binding is in gray underneath. And you can see that it's high in rutabaga and pebble to the right. They're closed in the adjacent neuroblast 7-4. And conversely, engrailed is closed to hunchback binding in 5-6 and open in 7-4. And the volcano plot shows about 100 genes that are differentially bound by hunchback. So we have 3,000 loci that are different, and hunchback's not binding all of those open loci, but rather a subset, maybe 10%, 15%. So hunchback is different and chromatin is different. And that very strongly supports the model of chromatin biasing temporal factor binding. And so the model that Sonia has developed and that her lab and our lab are still working on fleshing out, spatial factors act transiently to create neuroblast-specific chromatin landscapes. And those chromatin landscapes could lead to persistent spatial identity if they're maintained. That's an open question, an interesting one. Then temporal transcription factors such as hunchback show bias binding depending on which neuroblast they're in of the 30. And that bias binding leads to biased transcriptional output and the production of different neurons in each lineage. And so open questions that are very interesting for Sonia's lab and ours. Do spatial factors generate the neuroblast-specific chromatin landscapes? I laugh when I say that because really, what else could do it? But we haven't done any functional experiments. And Sonia in particular is looking at misexpressing and removing spatial factors and correlating that with chromatin changes to see if there's a functional relationship. But it really It's hard to imagine what else could be doing it. Do other temporal transcription factors besides hunchback show the same neuroblast-specific binding? We'd love to know if Krupel, PDM, and Cas bind. And I should make a little pro-user tip here, which is hunchback makes early-born neurons. Castor makes late-born neurons. All the genetics show that. You misexpress one, you get firstborns. You misexpress the other, you get lateborns. Everything says that. They're both zinc finger proteins, and Ward Odenwald's lab has shown they bind exactly the same DNA sequence in vitro. And so that raises an interesting conundrum that I would love to have somebody bite on and address in the lab experimentally is, how does the same DNA binding site create different early versus late when the proteins bind same DNA? And there's obviously many possible mechanisms. I'd love to know that. And finally, is neuroblast-specific chromatin maintained throughout the life of those neurons, especially the neuroblasts that are in the thorax? Their progeny persists through metamorphosis and participate in adult behaviors. And we'd love to know more about if they're carrying that spatial identity with them on their chromatin landscape. Okay, so that's the first story. So if chromatin put you to sleep, you can come back for the second story. And the second story is sort of a two-part story, both involving hunchback and asking, we know hunchback can create early-born molecular identity, but we don't know much else about that. And so the question now is, does hunchback create early-born neuronal morphology, synapse localization, and most importantly, connectivity? Because if it does those things, then looking for downstream genes of hunchback is the way to understand the mechanism of circuit assembly. Okay, this first Neuroblast 7.1 centric story is from Austin Soroka, a great graduate student who's now moved to fish and is not with us anymore. And his lineage was Neuroblast 7.1, and that's the canonical transcription factor cascade. And the neurons it produces are 5 motor neurons from the notch-on lineage, which innervates the motor neuropil. So that's where motor neurons are. So that notch-on hemilineage, there's U1 through U5, Eve positive motor neurons, and the first 2 are hunchback positive. Those are the 2 we're going to talk about right now. The siblings are interneurons, and we assume the same temporal patterning is being applied to them, but we don't have their molecular marks for interneurons. I'll come back to interneurons in the last chunk of the talk. Almost all of the temporal patterning experiments have been done on lineages that make motor neurons. So it's open question whether interneurons use the same mechanism. So to look at this lineage in a little bit more three-dimensionality, on the bottom, you can see an outline of the CNS. You can see a little volume box of a hemisegment. And Neuroblast 7.1 is at the ventral side on the bottom. Its firstborn is number 1, U1 at the top, and they're forming a diagonal. So the early borns are deepest, and then the later borns get pushed more and more superficial. But in addition, the neuroblast is moving laterally, one of the few cells that migrates in Drosophila nervous system. And so as it moves laterally, it's dropping these daughter cells off, forming this medial to lateral chain. So the chain of neurons is dorsal to ventral and medial to lateral. And we can use the fact that they're even skip positive and they're positioned to always identify their birth order in these experiments. Okay, so I want to talk about connectivity. They all are motor neurons. They all send axons out to the motor somatic muscles in the body wall. The 2 hunchback neurons in blue go to the dorsalmost muscles. The 3 nextborns that are hunchback negative go to the red lateral muscles. And so Austin wanted to ask, does hunchback play a role in directing this? Or as an alternative hypothesis, is it first come, first serve? The first one out, U1, makes it to the dorsal muscle and occupies that. The next one out can't get onto that muscle, takes the next muscle, and they tile because first come, first serve. And to determine if that's even a viable mechanism, Austin had to test to see if they all grew out sequentially or if they grew out simultaneously. And he did a little multicolor flip out experiment that's really elegant and convincing. And the data are shown in the middle as an example of one. This is a multicolor flip out in the Neuroblast 7.1 lineage, again, using a Neuroblast 7.1 specific GAL4 line. Thank you, Sunlin, for that 3 years ago for making that. And this multicolor flip out has 2 colors, 2 neurons marked in red and blue. The blue one is close to the midline, and so it's earlier born. The red one's further from the midline, more to the right, and later born. And you can see the earlier born one with the blue triangle projects out farther than the red one. So in this one example, the early born neuron is out there first, the later born is following. And if you look at the graph to the right, in any time where he has a pair of neurons within a hemilineage, which is about 10 of them, the one that's more medial or earlier born is always ahead in distance compared to the later born. So indeed, this model is a fair model on the upper right. They would go out at about 30-minute intervals into the muscle field. And so it raises the possibility that first come, first serve is the mechanism. The alternative mechanism is that they just go where their transcription factor tells them to go and connect up and form functional synapses based on these transcription factors and their downstream cascade of genes. And so the way to resolve this experiment experimentally is to misexpress hunchback. So now we have all of the neurons expressing hunchback, but they're still going out sequentially, 1, 2, 3, 4, 5 every 30 minutes, data not shown, or data published, but not shown here. And so the question is, where are those blue ectopic early borns going to go? Those later born hunchback positives, where are they going to go? Lateral muscles, dorsal muscles. And the answer is they go to the dorsal muscles. And so again, the little diagram shows the CNS with the muscle field adjacent to it. The little teeny box that you can't see anymore probably is showing what's shown in the confocal images, which is the lateral and dorsal muscle fields. And especially in the green solo image, you can see that these U1 through 5 neurons as a bundle labeled with the 7,1-gal4 driving myristoylated GFP form filopodial processes over both lateral and dorsal. The dorsals are the U1 and U2s. The lateral ones are the hunchback negative U3 through 5s. And as I'll show you later on in a couple of slides, these filopodia, which are not mature boutons by any means, but work from another lab that I'm going to mention in a few minutes, shows that they do turn into functional boutons. So I'll just give you that heads up that they're going to be functional. And the question is, what does hunchback do when misexpressed? And the answer is that these guys all go to the dorsal muscle field. You can see that in the muscle overlay, and you can see it very clearly in the lower green single label for the U1 through 5 neurons. They're all filopodia on the dorsal muscles, clean as could be in the lateral muscle region. Quantification is over there to the right. The bottom line of the quantification, which I can't really see from my perspective, is that in wild type, you have lateral muscle filopodia nearly 100% of the time. In hunchback overexpression, that goes to 0. Conversely, the dorsal muscle projections are more pronounced in the hunchback overexpression. And so the question is that a transient effect? And do they just compete and end up in the proper place an hour or two later because this is very early and they're just filopodia? Or do they mature and is this a long-lasting permanent connectivity? And are they even functional? And to answer those questions, your meeting co-organizer, Ellie Heckscher, in her lab at Chicago, had a graduate student, Julia Meng, who did exactly the same experiments as Austin was doing in a parallel universe right next door in experiment space, long distance away by physical space. And they found that they did the same experiment, except for they assayed at third instar larvae when everything is functional. And they assayed for functional connectivity based on GCAMP, induction of GCAMP signaling post synaptically. So the bottom left is their construct where they fuse GCAMP to discharge a postsynaptic protein. So now we're targeting GCAMP postsynaptically, or they're targeting it postsynaptically. And then the GCAMP signaling on the dorsal muscle boutons is done by summing GCAMP over 5 minutes because they're looking at minis, spontaneous release. So it's going to be a very low signal, but averaged over or summed over 5 minutes, and you get signal under every single one of those boutons. So that's wild type, and we expect them all to be functional, and they are. But in this experiment that Austin and Julia both did, misexpression of hunchback creates ectopic early borns that form functional boutons. All these boutons have increased GCAMP. They're all morphologically mature. And so what we can conclude from this is that not only do these late borns, when misexpressing hunchback, not only do they find the right muscle group, but they form functional synapses and increase the number of boutons on those muscles. So as it says in red down at the bottom, functional, and persistent. And so this is saying that hunchback is sufficient to trigger a gene expression cascade that will remap the circuitry at the neuromuscular junction, which is a very important result for motor neurons. Austin and Julia, for that matter, but I'll show you the data from Austin, wanted to look at dendrites also. So one of the first looks at within CNS morphology and connectivity. And so U1 through 5 have different dendritic projections too, so we can do basically the same experiment, but assay their dendrites within the CNS. And here we use multicolor flip out to identify individual U neurons. The staining is for even skip, so we know they're motor neurons. And then the distance from the midline tells us at what birth order they came from. And this is a control. And so U1, the cell body in the little inset below shows even skipped in the neuron that's right next to the midline. And so that U1 that I'm showing you there has positioning of its cell body in a U1 morphology spot right next to the midline. 2 away from the midline is where U2 is found, 3 away from the midline, U3, 4 away from the midline, U4, 5, U5. This is wild type. You can see that the hunchback neurons are morphologically different in the CNS. They have a bipolar U morphology, and they have contralateral, ipsilateral and contralateral dendritic arbor. Whereas the later borns that they don't cross the midline, and they only have ipsilateral arbor. So does hunchback play a role in arbor neuropil targeting? Can it control where neurons are going to send their axons within the complex 3-dimensional neuropil? And the answer is yes. So when we misexpress hunchback and look 1, 2, 3 out from the midline, we can give them names. U1 and U2 are totally normal as expected because they were already hunchback positive. U3, 4, and 5 are now named U1, 2, 1, 2, 1, 2 because they've been transformed. They're not their normal morphology, but their 3rd, 4th, and 5th position off the midline. And the little graph in blue off to the end shows how many of these, what percent of these later born cell types actually produce a contralateral dendritic arbor in the right place. And you can see that U1 and U2 always are 100%, and then you fade down. But even the 7th born one, there's an example or two of a 7th born that's like 3 hours later than the normal U1, and it still has a contralateral, which up and to the left dendrite. You can't see the midline marker because it's too faint, but all of these neurons have a contralateral, that means on the left side, dendritic arbor. And so that's really remarkable. They don't get there by the same route. U3, 4, and 5s are not bipolar. They send an axon up like they would normally, and then form a dendritic arbor like they would normally, motor neuron like they would normally, but then they send this weird contralateral projection that goes exactly where it should go on the other side. So they're taking the wrong path, but they're getting to the right place. And that's true even for the 7th born in some cases. And so the conclusion there is at the bottom, temporal transcription factor identity, i.e. hunchback, determines dendrite morphology, putting those dendrites in the right place. And if we look with a beautiful multicolor flip out example from one of the segments that Austin looked at, you can see an endogenous U1 in green. We know it's U1 because it's right next to the midline, the white dashed line, and has the U morphology. The ectopic U1 in red is off the midline, so we know it's later born. It has a T-shaped morphology, so we know it's transformed, and its projection is shown in the enlargement there. It's intensely, tightly interdigitated with the normal U1. So these ectopic rewired dendrites are going to precisely the same volume as they would normally go if they were a U1 or a U2. So this really shows that whatever hunchback is doing, it's allowing these neurons to read the neuropil in a way to find exactly the right volume to put their presynapses, postsynapses. Okay, so open questions on Austin's project and Julia's project. What maintains neural identity after transient hunchback? So hunchback, like the spatial factors, go away, so too does hunchback go away. How is it temporal fate maintained? Does it trigger a combination of homodomain transcription factors like Oliver Hobart suggests in the worm? Does it address change chromatins? Now we've got 2 different sequential chromatin modifications that lead to a permanent spatial and temporal identity. These are all very interesting questions. And we're currently doing a lot of work on hunchback on single cell RNA-seq, lineage-specific single cell RNA-seq to try to tease out who are the downstream components of the hunchback pathway that create that beautiful axonal connectivity and dendritic morphology. Okay, in the last little bit, I want to tell you about totally unpublished work, mostly from Heather Paulington, a new graduate student, but based on some transmission electron microscopy work done by Brandon Mark. This is a new lineage, neuroblast 5-2. It's not 7-1, which we know so well. Its firstborn neurons are interneurons. It has at least one firstborn interneuron called AO6K, shown in blue up there. And the point of studying this lineage for Heather's sake and for my sake is, does everything we learn from motor neurons translate over to interneurons? And can it rewire and trigger appropriate connectivity in a totally different type of neuron, the interneuron in the CNS? Another reason we're interested in 5-2 lineage is, again, work from Ellie's, not Ellie's lab. This was when Ellie was in my lab. Work from Ellie when she was in my lab showed that the late-born neurons are part of a proprioceptive circuit, which in her lab, she's really done a beautiful job characterizing. But her early work showed that the proprioceptive circuit was composed in part of those late-born neurons. And so we already had some circuit data, and we wanted to see if we could add into it by putting hunchback into the mix and seeing, A, does it create interneuronal diversity? B, does it trigger them to wire up with other lineage-specific neurons? And we don't have the answer to how they wire up yet, but I will tell you about their role in specifying interneuron identity over the next few minutes. Okay, so neuroblast 5-2, to study it, we need a neuroblast-specific GAL4 line. Haluk and Senlin came up with another beautiful neuroblast-specific GAL4 line in neuroblast 5-2. The pattern is shown in the middle image in green. The projections into the brain are less clear, cut off in the volume. And you can see that it produces a segment-specific lineage with contralateral and ascending neurons. Brandon went in and took EM volume done by the Cardona lab and annotated every single neuron in the A1 segment. And I mean, not every single neuron in the 5-2 lineage in A1. And so we have ground truth data from these neurons that have been EM reconstructed. What are their morphology? What are their connectivity? And now we just have to figure out which ones express hunchback and start studying them with our same tools. And so I'll zoom in on A1. On the left is the confocal image of the two lineages with their projections. On the right is the fully reconstructed lineages that Brandon found at newly hatched larvae in both cases. And you can't really see much structure. You can see some contralateral bundles, but the structure and the projections become really super apparent when you rotate 90 degrees and look end on. So now we're going to rotate these guys and the little diagram, rotate 90 degrees. And now we can really start to see some diversity in projections. The lineages are shown with their cell bodies on the bottom. They project into the neuropil. And there's a ventral commissure, you can see, a dorsal commissure, and a single neuron that forms an X in between those two commissures. And you can see that if you're lucky in the first couple rows in the EM reconstruction, but trust me, it's there. And it's a little bit more visible in the confocal. And it turns out that Heather did multicolor flip out in this lineage, mapping and standing for transcription factors. Hunchback is what I'm going to tell you about today, Krupel, PDM, and Cas as well, to try to figure out which of those neurons that's in the EM reconstruction is expressing hunchback, Krupel, PDM, or Cas. And she found the hunchback neuron. It's the one that does the little X in the middle, which is great because that was a very distinctive morphology. And hunchback positive neurons typically have quite distinctive morphologies in other lineages as well. In the EM, you can see the left and right sides of this AO6 interneuron cross at the midline in that beautiful X. They have presynapses to the lateral side of them, sort of centrally in the neuropil, and then projections up into the brain, which I'm going to ignore for the rest of the talk. Heather's multicolor flip out shows a cell body in the same position, dendrites with their, and some presynapses in the middle going up across. This is unilateral, just one, so you're only seeing half of the X, and then up and out to the brain. And down below in the inset shows it's a hunchback positive neuron from this MCFO. So now we have a hunchback positive neuron, interneuron, and we can ask, can hunchback specify later born neurons into this interneuron identity? And does it have the same interneuron morphology? And at some point we haven't gotten to, connectivity. And so this is the same control I showed you before. Keep in mind the dorsal, the ventral, and the X projections. Dorsal and ventral projections are from late-born neurons. If we misexpress hunchback, we expect them to be diminished. If hunchback is remapping neurons, we expect that X to be more exaggerated as more neurons take that morphology. So that's what we're looking for. That's our expectation based on motor neuron data. And the answer is that's what we see with the interneurons too, a really profound transformation in their morphology to an earlier born type of morphology, this X neuropil crossing and projections out to the brain. And you may think that the segment on the right has fewer cells and there's cell death, but actually quantitatively for this experiment and the next one I'm going to tell you about, for everything we've done in this lineage, there's no cell death. There's no apoptosis. The cell numbers are identical. So it's just the way the image was taken didn't include all of the cell bodies on the experimental. But you can see that beautiful X. And so the conclusion from this experiment is the hunchback specifies interneural morphology in the same way it's specifying motor neuron morphology. It's changing late-born projections to match early-born projections. So it's controlling the gene suite of cell surface molecules and transcription factors. Whatever's downstream of it is important enough to remap the projections of this neuron. The final experiment that Heather did that I can tell you about is to look at presynapses, BRP positive puncta that label presynapses to see if synaptic targeting has changed. I showed you those neuromuscular boutons from my work in our lab and from Ellie's work with Julia, how misexpression of hunchback led to ectopic boutons that were persistent and functional. We wanted to know if the same thing was happening here. The prediction then, this is a 5-2 specific expression of UAS BRP, a presynaptic marker. And in the EM reconstruction cross-section, what's shown in the cyan box is where the presynapses are from this interneuron. And what's shown in the magenta box is where some of the later born neuron presynapses are. And Heather has quantified these two boxes. It's new data, and she hasn't quantified the other presynaptic sites that are also from later borns more dorsally, just because it's such a laborious job. And so this is the first look at this data. And we would predict based on motor neuron data that we would lose those late-born synapses because they're being transformed into early-born X-shaped neurons, and there would be no projections to their normal locations. There would be no synapses there. But so we expect them to be lost or reduced, and we expect synapses density to be increased in the area of the gray, in the cyan box. And these measurements Heather takes are very precisely stereotactically quantified. So X% down, X% AP, X% medial lateral. So she's measuring the same volume from segment to segment, from embryo to embryo. And what she found at a qualitative level is up in the upper right is a loss of those late-born presynapses. That was a very much expected result, just like we lost the projections of the motor neurons to the lateral muscles that never recovered. Here we lose those presynapses because those neurons are in a X-shaped morphology and they don't even go to that part of the neuropil. But it's impossible to tell by eye, just looking if there's more in the early born synaptic volume. And in fact, there isn't. If you look at the quantification down below, the top cyan bars show a statistically insignificant number of presynapses. So unlike the motor neuron experiment where you make a bunch more neurons projecting to a particular muscle target and they stay overproduced in too many boutons all the way to third instar, here at least it looks like there's something else going on. The theory that we have right now is that they're compensating, so you're sending too many neurons there, but each neuron forms fewer synapses because there's some sort of compensation. And it's not a case where you're having too many neurons with the same number of synapses like we do with motor neurons, which would create an overabundance of synapses in that volume. And we definitely don't see that. So the conclusion from this experiment is that hunchback determines presynaptic targeting in the sense that it takes those presynapses that were in the late-born neurons and moves them into the central domain where the early-born presynapses are. The other possibility is that those neurons are still there, but they're not making any presynapses. It's just the endogenous ones. And this will have to be teased out with multicolor flip-out experiments. Okay, so Open questions here. Again, what are the targets of hunchback in this lineage? We have done quite a bit of, in the old days, microarray experiments where we looked at wild type versus hunchback misexpression throughout the CNS. We never could find anything that was really useful or interesting from those experiments. Now, looking at hunchback specific functions in each of these lineages, we know that that was a messy experiment. And now we're going back and doing single lineage RNA-seq to see if we can figure out hunchback genes that correlate with hunchback expression, whether it's in wild type or misexpression. So what are the targets? That's an active area of investigation. And does hunchback specify connectivity like it does in the muscles? We know they target like they do in the transformed late-borns target properly, but do they form functional connections? And are those connections part of the proprioceptive circuit that Ellie described so beautifully several years ago? So those are open questions. So in conclusion, I've told you that there's 3 steps to getting a neuronal diversity produced. The first 2, spatial factors, control progenitor identity. And they do that, we think, by specifying different chromatin states in each of those 30 neuroblasts. Then temporal cue, the same suite of temporal cues come into each neuroblast and give different results because they're acting on different chromatin. And that creates 150 different cell types. And then I haven't had a chance to tell you much about the notch on, notch off final division, but that one, of course, brings it up to 300 cell types per hemilineage. And the open questions that are sort of big global open questions, what genes are regulated by hunchback and all the other temporal factors? Do they all play by the same rules, or is hunchback being first in some way different? And that's just a really important question that we need to dig into, is doing the same experiments on the later transcription factors. And finally, the question that I'm sort of most interested in, because I just think it's really cool, we've shown chromatin differences in neuroblasts. It's possible that the temporal factors trigger chromatin differences because they're only transient as well. And hunchback is known to interact with DMI2 and be involved in chromatin changes at the Hox loci. So there's some evidence that maybe there's another series of chromatin modifications. And then, of course, if notch signaling triggers chromatin modifications, you could end up with 300 neurons that are all different at the level of chromatin instead of transcription factor codes, for example. So assaying for chromatin state at the next 2 mechanistic levels is high on our list. And I'd like to thank the people in the lab on the right that did the work, those 5 people, the people that shared reagents and data, especially Julia and Ellie, for making a potentially competitive situation into a pleasurable, coordinated publication system. Jim Truman, the Genelia Fly spaceship, and funding agencies. And again, you guys know there's no questions, but I'm going to be down there with a beer. So I really love to hear if anybody has any questions. Thank you very much.